Thank you all for staying and for hopefully getting enthusiastic about what we're going to talk about, which is new approaches to generate evidence for treating pregnant and lactating persons. As I've been sitting through the meeting the last 2 days, it's been a tremendous meeting, actually. I've really enjoyed it a lot. It's been really interesting to think about because I'm on some of the generating evidence side of this discussion, how I can see huge potential for the opportunity to start to fill in some of the gaps that we've identified. And you'll hear about that from my colleagues on the panel today. So I'm Steve Kern, and I'm Deputy Director of Quantitative Sciences at the Bill and Melinda Gates Foundation. What that means is I lead a group that basically works across all our therapeutic areas to figure out how do we better inform the studies that we're funding, the projects that we're working on, how do we integrate the data. We do a lot of PK/PD modeling across therapeutic areas, and I've been a constant advocate for not leaving pregnant women out of our studies, particularly in anti-malarials, where, as we heard earlier, they can be a real problem. I'll apologize to you all for I don't have a tie today. So, this is only my second post-pandemic trip. So when I got to my hotel, I opened my bag. I was like, forgot a tie. Okay. And the bookshop's not open. I was trying to get one from the bookshop. So I'm not usually this casual in Washington, D.C., but anyhow, thank you. So I beg your forgiveness. So we're going to have a really great session with presentations from 4 different folks who have been working on both technological and experimental aspects to start to fill in some of the pieces of information that we were all hearing or missing. And so I'll really want to turn this over to that group. And so to my left, I have Dr. Tina Chambers from UC San Diego, and she co-directs the Center for Better Beginnings and is program director of the Mother to Baby program that builds evidence based on information of exposures during pregnancy and lactation. And then also is leading the Mommy Milk Human Milk Biorepository for research, which we were just talking about. It sounds like a phenomenal resource. And she's going to talk about real-world evidence perspective, but in a very broad sense of real-world evidence, not just thinking about EMRs. And then to her left, Dr. Raman Venkataraman, or Venkat, who's a professor of pharmaceutical sciences and pathology at the University of Pittsburgh. He's going to talk about pharmacology perspective and talk about a lot of the tools like physiologic-based pharmacokinetic modeling, or PBPK. These are the things that actually really help the pediatric program make great strides, and so he'll talk about some of those in this context. Next to Dr. Raman is Dr. Tolua Lashe, and she goes by Lashe Ajayi, and she directs the clinical research and diversity initiatives at the Scripps Research Translational Institute and is an assistant professor of pediatrics at UCSD and the Rady Children's Hospital, where she's a hospitalist and a pediatric palliative medicine physician. So both research experience as well as hands-on clinical experience. And then last, joining us virtually, Dr. Wei Hao, from the FDA, who's acting deputy director in the Division of Epidemiology and the Office of Surveillance and Epidemiology, who will give us a regulatory perspective. So each of the members of the panel will talk for about 5 minutes, and then we'll save the rest for discussion and questions, hopefully from the audience as we get forward. So with that, I'll turn it over to Dr. Chambers.
Thank you, Steve. And I didn't wear a tie today either.
Okay, thank you. We both wore blue blazers, so that's good. Okay.
So I want to thank you and the organizers for the opportunity to take part in this workshop, a topic near and dear to my heart. So as we all know, there's a variety of real-world data that are used to address safety and potentially effectiveness of medications in pregnancy and, to a lesser extent, probably lactation, including electronic health records, claims data, and pregnancy registries. I'm involved in generating real-world evidence through 2 separate but related research initiatives centered at UC San Diego. And the first is based on our National Counseling Service for pregnant and lactating persons, Mother to Baby, that's built on a network of about 14 services, first established about 40 years ago to provide evidence-based, individualized risk assessment, so counseling to pregnant and lactating persons and their healthcare providers about exposures that they may have experienced or are planning on, medications, vaccines, and other environmental agents. As you've heard, more than 80% of medications have limited or no human safety data with which to counsel, so we struggle with this every day. And for that reason, we use this platform, the counseling service, as kind of a natural springboard for inviting pregnant women to enroll in longitudinal prospective cohort studies or a pregnancy registry on the basis of them already having had a given exposure or exposures. And at the same time, we have the opportunity to enroll comparison women who have contacted the same service but have not had the exposure of interest, but may have the same underlying conditions with different or no treatments in pregnancy. So we follow these pregnant persons and their infants through pregnancy and to outcome to at least 1 year postpartum, with consent to recontact for further follow-up as necessary through 10 years after the end of the pregnancy. And we collect information on exposures and outcomes through telephone interviews directly from the mother. She's the one who consents. Medical records abstraction from all of her providers and hospital. And in most cases, we also offer a specialized physical examination for live-born infants that one of the study pediatric geneticists or dysmorphologists conducts. We used to do these in their homes, but we now do them by telemedicine. And then many of the studies involve standard neurodevelopmental follow-up of the children through the first year of life and some to 5 years of age. So we currently have projects that are funded by industry or federally funded. The industry ones fulfill post-marketing commitments or requirements, and we have about 25 different products under study. We also collaborate and have done so since 2009 with the American Academy of Allergy, Asthma, and Immunology on something called the Vaccines and Medications in Pregnancy Surveillance System, or VAMPS project, which brings together our cohort arm, or the pregnancy registry arm, with a complementary and parallel claims data study and a case control data collection arm, all under one roof. So these are conducted in parallel. So using real world data from 3 different study designs. And then on the lactation side of things, we established a human milk research biorepository, as Steve said, called Mommy's Milk, the public-facing name, in 2014 at UC San Diego. So we enroll lactating persons throughout the U.S. and Canada. We recruit through the mother-to-baby counseling service and pregnancy studies, but we also, a high proportion of women are recruited through direct-to-consumer social media advertising. So each mom provides a detailed list of her medications and all of her other exposures over the previous 2 weeks. She provides breast milk sample or samples, clinical data from maternal interviews, and she releases medical records for herself and her child annually, and also participates in neurodevelopmental follow-up. And this effort is in part funded by federal funding through the NIH. So Diana Bianchi talked this morning about the MPrint Hub. So we're a center of excellence in the MPrint Hub and also take part in the Pediatric Trials Network CUDDLE study. And we have some industry-sponsored studies, for example, looking at remdesivir in breast milk. So having both the infrastructure for the pregnancy and the lactation research platforms in place on an ongoing basis with real-world evidence has really allowed us to tap into exposures of concern or interest and to respond quickly to emerging needs. So as we've talked about earlier today, the issue of COVID-19 and COVID-19 vaccine came upon us relatively quickly. So having that infrastructure in place was invaluable in being able to pivot at the beginning of the pandemic to enroll several thousand COVID-19 infected pregnant and lactating women and uninfected comparison women starting at the beginning of the pandemic, and then starting in December 2020, lacking data from clinical trials at the time of emergency use authorization for the vaccines. We've been able to enroll several thousand vaccinated pregnant and lactating women with COVID-19 vaccines in comparison women who had declined to be vaccinated. So these two interrelated platforms from the real-world perspective, we think can kind of serve as a surveillance system. So there are many challenges, and these are primarily, as was discussed in the breakout sessions yesterday, awareness and incentives for clinicians to refer or women to participate. And this is assuming that the exposure of interest is actually occurring in pregnant or lactating women. But the goal is for us is to find the best ways to make sure that the women or provider know about the study. And then our second challenge is diversity. And I think we're going to hear a little bit more about that as we all struggle with issues of who is it that learns about a study, who gets referred into it, who consents, and they tend to be not a very diverse sample of participants. So ideally, from my perspective, if there was one initiative in the whole pregnancy registry space, we would move towards supporting a single pregnancy registry model in the U.S. It would be extremely more cost efficient, way less complicated than what we do now. And this one population-wide pregnancy exposure registry could be capturing data on all new medications. So every medication, as soon as it's approved for use in the general population, when pregnancy exposures occur, it would simplify from the clinician's perspective and from the patient's perspective how they would become involved in such a study. Talking earlier about opt-in, opt-out, and of course could be used to target existing medications of high priority. So in terms of how this fits into the array of approaches that might be used to study medication safety in pregnancy and lactation, I think this is complementary to a move towards doing more randomized clinical trials as the gold standard. However, real-world data will continue to be necessary to complement clinical trials, in my opinion. So clinical trials, even if focused on a pregnancy treatment for a common underlying condition or a vaccine, are going to be challenged to be large enough to address risk or safety, especially with respect to first trimester exposures and rare outcomes such as congenital anomalies. So you're going to need other approaches to try to address those outcomes. You'll still need large cohort and case control studies. And then moving forward, I think we have lots of opportunities that have been discussed here already. Formal and structured collaboration across different data sources in the U.S. and globally, formal processes for identifying priorities, sharing data and findings, and sustainability. Standardization of data collection, both in terms of exposure and outcomes, allowing cross-platform and cross-study comparisons. And I think a model we might look to that's been mentioned several times before is this EU Conception project that's aiming to create a platform for data collection, analysis, prioritization, and knowledge sharing, and it's one that we could emulate in the U.S. Thanks.
Thanks, Tina. And we'll go now to Venkat and then keep going on.
Thank you, Steve. Good morning, everyone. When we consider pregnancy and drug, we need to think in terms of it as a twin issue, pardon the pun, two separate components. One is novel agents used for pregnancy-related, induced, or mediated conditions, such as preterm delivery, preeclampsia, etc. The second one is more how do we use approved drugs which are necessary to treat pregnant women when they happen to have either a chronic condition or an acute condition for which we need medication. My initial comments are going to be focused on the latter or the second aspect of it. There are 5 components we need to keep in mind: efficacy in mother, safety in mother, safety in the fetus, neonatal safety, and efficacy in the fetus in special circumstances. So I'm going to focus predominantly on the efficacy and safety in the mother to begin with. In any pharmacotherapy, the ultimate goal is to optimize the dose of a drug to a given response. And so if anytime we can measure a response and titrate the dose, that's what we need to do. But what is the limitation? We can't very readily or easily measure drug response in most of the cases. When we can, it's too late. And so what is the solution? Exposure of the drug is a surrogate marker. In other words, if we can make the assumption that similar exposure leads to similar response, then we can look at the exposure and infer what is likely to happen to the response. So I'm going to keep the theme of what is the barrier and what is the limitation. So now let's look at exposure. Ideal way to look at exposure is to look at what's called the area under the plasma concentration time curve over a given dosing interval. What is the limitation? It requires the mother to be in a clinical setting for 12 hours, plus take multiple blood samples, not always very practical. So what may be the solution? Well, if we really need the 12-hour sample, we need to make sure that there are opportunities provided for care to the provider, support system, somebody to come with them, somebody to take care of their another baby that they may have, and some transportation and access and other cost. Other solution is something called a limited sampling. In other words, instead of taking 12-hour sampling, we can take samples, 2-3 samples in the early part of the plasma concentration time profile and make an inference on what should be the theoretical, the exposed AUC. Or we can look at the trough concentration, single measurement. But we need to document that trough concentration, and the limited sampling, in fact, reflects the entire area under the curve. What are other new approaches that can simplify patient issues? There are novel approaches for blood sampling called something based on a dry blood spot. It's an advanced technology called volumetric adsorptive measurement system, where it is like a Q-tip. The patients can sit at home, just like taking a blood glucose level, prick, push, touch, put it in an envelope and send it to your lab. That system is being evaluated, and that's likely to minimize the inconvenience to the patient. If safety becomes a concern in using any medication in a patient, microdosing is perhaps an opportunity. Then there are approaches that we can use, such as population pharmacokinetic model, where we have limited sample from a given patient, but we have that limited sample in a large number of patients. We can consider all of this having come from one person and really model it, and eventually look at what are the various covariates, cofactors from the patient that makes that data point deviate from the model predicted values, and therefore infer whether pregnancy contributes to the observed variation, identify pregnancy as a cause of variability in the exposure. There are other opportunities like placenta and a chip. Here we could potentially look at simulating concentration profile by adjusting the flow rate, adjusting the concentration, and therefore look at in vitro pharmacokinetic simulation, look at safety issues. We could do a placental transfer and see what drugs may potentially transfer, what drugs can impact the placental cells. The next area is we don't know everything about physiology of a pregnant woman. We know about cardiac output, GFR, albumin, and hematocrit. We don't know about all the CYP enzymes. We don't know about all the transporters. We have to systematically study them using some sort of a probe medication. Kathy mentioned about this, and I think those studies are valuable studies to do. Once you have that probe assessed in a population of pregnant women, take that information, feed into the PBPK model, and make the model predict things much better. Models are only as good as the assumption and the data that goes into building these models. We need to keep that in mind. What if we don't want to give any medication in order to know what happens? Biomarker. There is potential for some biomarkers which may be endogenous, which may tell us something about liver function, tell us something about the kidney function, indirectly therefore imply what kind of a dosing we need to give. the other factor we need to keep in mind is that all of these things only give you an average change. It tells you twofold difference, threefold difference. It doesn't tell me or anyone how much change you have versus how much change you have. You may have 10% change, you may have 300% change. So we need to understand what factors. It could be genotype, it could be hormonal levels in the particular patient, it could be cytokines and pro-inflammatory mediators which modulate the enzyme activity. All of those could contribute to that. But ultimately, there are studies which have also shown that PD, or the pharmacodynamic response to the drug, does change. Especially one of the OPRU studies documented gestational diabetes. The insulin sensitivity is different in gestational diabetes compared to the type 2 diabetes. So in other words, we need to make sure, anytime possible at all, do pharmacokinetics or collect data. Ultimately, if we think we have limited data in pregnancy, lactating women, we don't have a data. Lactating women are not pregnant women. Physiologically, they are very different, and we need to systematically start doing studies in this patient population, not only from the mother's perspective, but also from the baby's perspective. That leads to the last component, lactation studies. Ideal lactation study is to have the mother pump out the milk over a dosing interval, look at the total amount that's excreted in the breast milk. Therefore, we know the cumulative potential dose that is exposed to the fetus, to the neonate. But it's not very practical. Many of them may not want to not feed their baby. So the next best alternative will be taking samples, simultaneously taking plasma, blood and milk sample. Even if that's not possible, just take at least minimum one sample close to the maximum concentration. Make some assumptions. What is the average milk production? And calculate the highest potential dose that the baby can be exposed to. So these are alternatives. Last, I think the problem we have is we generate data, we make observations. It takes a long time for it to put it to practice. We have got to condense that time span. I think associations, professional organizations must take a lead in translating clinical observations in research setting to practice. Thank you.
Thank you, Venkat. Lashe?
Thank you so much. I became pregnant with my second daughter in June of 2020. I was in my second trimester when the COVID-19 vaccine became available to clinicians in the hospital. At this time, we were at the height of the pandemic. I was shifted to the adult palliative care service because of how busy we were. I knew my risk was high. I was at the bedside of patients dying from COVID-19 because families were not allowed to be with their loved ones in the hospital. I was the one there at the bedside. And yet, I wasn't sure what I was going to do about the vaccine. I asked my OB. At that time, she didn't have enough information to really give me a recommendation. I'm pretty educated, clinical researcher, physician, yet I had my doubts about the vaccine. I didn't trust the administration that the vaccine was coming under. I was nervous about it. I'm a Black woman. I still have those preconceived notions. Luckily, I have access to my friends from ACOG who advised me. I got access to the data, and then I did it. I got the vaccine, but more than that, I went on social media, on Twitter, actually posted my experience getting the vaccine at this time now in January of 2021. And I went on social media and I documented my experience from getting the vaccine. I put myself and my body out there. I gave my medical history. I talked about my fibroids. I gave my labs. And I documented everything that happened throughout my pregnancy and when my baby was born in March of 2021. Also, during this time, I became the lead researcher in a mobile research platform called Powermom. And my experience and my journey being pregnant and getting the COVID-19 vaccine at a time we didn't have any information, and just how viral probably the only viral tweet thread I'll ever have in my life. When I told that story, it really changed how I approached this research platform when I became the principal investigator. So what is Powermom? Powermom is a mobile research platform. It's bilingual. It's survey and wearable sensor based that incorporates EHR technology or information to really have the goal of having the largest, most diverse pregnancy and postpartum research community out there. Our goal is to engage 1 million pregnant and postpartum people and engage them in clinical studies, observational, to get more information about pregnancy, to support this community, and also provide information. And as we get to our goal of 1 million, to really then use predictive analytics to inform our research community and share information. Why does that matter? Why does that experience matter to this group? Because I've learned that when you use a platform to lift up the voices of people who've been left behind in research, not just pregnant and postpartum people, but also people of color, you get this rich, diverse database that can be used to inform all of us. How are we able to do this? The first step that I learned from my lived clinical and just, being a person and pregnant person experience is that we have to create and develop the research with the people that we want in mind. So Powermom is directly driven by and informed by our participants, right? Obviously, I work with my OB colleagues. Again, I'm a pediatrician and adult palliative care physician, but I work with my OB colleagues to make sure the information I'm gathering has clinical relevance and is important. Then how we conduct the study, how I use this mobile research platform to make sure that the people that I want to engage in research use it. I work with them to have them drive how we time the questions, how we time the surveys, and how we deploy the information. They built it. They give impact or information about how it's built, the look and the feel. And I work with community advocacy groups. One of my sponsors and partners is March of Dimes. I work with Microsoft. I have input from legislators. So it takes a collaborative village, and all of them provide that input because to do this research, to do this well, I need input and to be driven by the many partners that are active in this space. Next, I make sure that working with this group, the participants and our community advocacy groups and our national partners, that we are bringing the research to them, right? As we've said so many times, pregnant people are people. We're busy, and sometimes being pregnant just means that you have a second full-time job on top of it. So then making our research fit their lives rather than making them fit our research protocols. So we, again, make it so it's accessible to them and they can answer the questions within time frames that we need it to, but so it fits their schedule and not our schedule. Lastly, how we disseminate the information that we get. So, of course, with our different subsidies that we have that allow us to, go deeper into some of the pharmacological studies that pharmacy questions or mental health or different subsidies that we have. But then we use the mobile application to also disseminate information. When you answer a question about, hey, who got the COVID-19 vaccine? As we get more funding, we can actually show X amount of people just like you also got the COVID-19 vaccine or also taking this medication. So it builds that community in that real time. They own their own data, right? They choose what they share with us, so they have access to it and they own it. But then they also get to see in real time how their information is contributing to science and impacting other participants just like them. So I feel like the lessons that I've learned within my experience and also what I learned from the participants within Powermom is to really how to truly conduct participant and community-focused research to directly meet their needs. We have this infrastructure to collect opt-in pharmacological studies. We have this platform right here. We have an engaged population, and I think lessons from Powermom can really inform a lot of what we've talked about here today. How to engage this population, how to give voices to those who don't have a voice, and really how to share that information in real time so they get engaged and they stay engaged within this platform. Thank you.
Thank you, Lachey. That was really interesting to hear about and really relevant for your own personal experience, obviously, as well. So last in this, in our presentations, and then we'll go into discussions, is Dr. Wei Hao from FDA. And Wei, can you hear us okay? I can see you on screen.
Yeah, I can hear you. Can you hear me okay?
Sounds great. Go ahead. Sounds great. Go ahead.
Okay, that's great. Okay, thank you so much for having me here today. I think that the discussions in the past 2 days have been very much helpful and interesting. So talking about the new approach, I would like to share a few points from the regulatory perspective. And so this is my disclaimer at the bottom of the slide. So next slide, please. So while it's challenging to include pregnant women in clinical trials, the FDA does have the authority to acquire post-market safety studies under the Federal Food, Drug, and the Cosmetic Act. The regulation authorizes the FDA to acquire studies at the time of approval to assess a known serious risk or a signal of serious risk, or to identify an unexpected serious risk when data becomes available to indicate the potential for a serious risk. And such studies can also be required after approval if FDA becomes aware of new safety information. Next slide, please. There are 3 general approaches in the post-marketing setting: pharmacovigilance, pregnancy registry-based studies, and the pharmacoepi studies using complementary data sources. Pregnancy registries remain an important tool in the post-marketing setting because of the prospective design and the ability to collect detailed patient-level data. However, because the patient recruitment and the retention are often challenging, pregnancy registries are generally not sufficient by themselves. On the other hand, complementary studies with electronic healthcare data such as EHR and claims data may help address the limitations inherent to a registry-based study. Next slide, please. However, to achieve quality evidence, there are still a lot of scientific and technical issues to be worked out for studies using electronic healthcare data. It should be noted that a wealth of methods have been developed and validated in the literature, which is very exciting. But the concerns are still there because the validation approach and the results are usually not optimal. So in general, the data and the methodological challenges differ by the purpose of study, the study question, type of medication. This relates to the disease area, the anticipated magnitude of exposure in pregnancy, the benefit and risk, and also the nature of the outcome to be investigated, the characteristics of data source, the type of study, etc. So the issue, the impact should really be evaluated on a case-by-case basis. Rigorous studies are desired to inform regulatory decision-making. Next slide, please. So given that, and there are a lot of ongoing efforts, FDA continues to evaluate and develop methods and data sources to improve the capabilities of drug safety assessment in pregnancy. The FDA Sentinel system is a congressionally mandated distributed data network for drug safety surveillance. So one focus of the Sentinel system is to define and redefine the methods to assess medical product utilization, safety, and effectiveness during pregnancy. In addition to Sentinel, we have also collaborated with outside research groups to develop the capability of using the national Medicaid data for post-marketing active surveillance to assess pregnancy safety. Further, to fulfill the PDUFA 7 pregnancy safety commitment, FDA expects to have the improved capabilities in a consistent approach to pregnancy safety PNRs and PNCs by 2027. I think this concludes my 5 minutes. Back to you, Steve.
Great, thank you very much. So this is great. It was really interesting, I think. Let's give way. Thank you. You know, I think one of my takeaways from listening is just how the idea of maybe perhaps integrating a lot of the pieces that we just heard from our discussants could be really powerful to filling in some of the gaps. But I want to open it up for questions, and ask Deanna, is there anything on the virtual, so we keep our virtual audience engaged?
Yeah, so we have one that says, what are some innovative approaches for carrying out longitudinal studies that include pregnant and lactating people and could account for long-term impact on maternal or fetal health, for example, through puberty or even to fertility?
Oh, wow. So maybe, Tina, if you want to start off with that one, if you're going to go beyond 2-year-olds and 5-year-olds to pregnancy and maternity, and then maybe go to Lachey next.
I think it was mentioned by someone in the last 2 days that when you talk to a pregnant woman and you're talking about risks, that she cares a lot whether or not the child has a congenital malformation, but she really cares a lot how well the child's going to do in school. So I think parents are motivated to want to be able to capture that information. So the challenge is, if you've engaged them early on, to be able to retain them over the course of the period of time that it would take to collect that information. But I think there is a value in doing it, and I think it is possible to do it, and we've certainly succeeded. I mean, we have a really high rate of retention in our pregnancy studies to 1 year, about 95%, but we do I think we really can maintain pretty close to that with retention long-term for those that are willing to participate in the long-term neurodevelopmental and other outcomes, and it's definitely an endpoint we should care about, and we should set in motion ways to capture it.
Yeah, I really want to echo what Tina said. And, our study only goes to currently 8 weeks postpartum, but the innovation to engage in retention is to honestly provide information that the participant cares about. So what we do is that we continually add information as it comes out, articles about COVID-19 and pregnancy, information about exercise and development, information about going back to work, breastfeeding, pumping, things of that nature. And so really information that uses a tool that they look in front of every day. I think all of us in this room have a smartphone, right? We also know that really 90% of people in childbearing age own a smartphone device, including that's 86% of those who make less than $30,000 a year. So we have a tool that actively captures this population. It's how we utilize that tool and how we provide information that's relevant and pertinent to them to keep them engaged and coming back to your research site, your research platform.
Thanks, that's great. And actually one of the most impactful patient-driven organizations that I've seen have been a group, actually it's a very rare disease in mostly women who self-organized around data on their cell phone and used their cell phone as a platform to share data among other women that had the same condition. And now they are an incredibly powerful patient advocacy group that sponsors their own research. They even supported the indication of a drug off-label for use as treatment in their own disease. So it can be tremendously powerful if we can help bring that capability, as you say, to the patients where they are. Is there a question in here, perhaps? I get you just to talk into the microphone. Thanks.
Out of curiosity, do you find that the availability of free publications that literally wind up in every household, such as Reader and Penny Saver, that literally do wind up in every household, do you find a greater participation in the trial studies than other areas of the country?
Wow, that's a really interesting question. I don't know if anyone has a perspective on that in terms of, how are you finding women into your studies that you're doing, Christina or Tina?
So we get them from the counseling service, so they are already interested. So they've contacted us, and they already, or their provider has referred them, so they already know that they have a question about this. But over the last 10 years or so, social media has become a huge resource for us. And so a high proportion are self-referred now who learn about this through social media and then contact us. So I think social media is our penny saver of the 21st century.
I would add to that, honestly, using other pregnancy applications that already exist. Right now, we're able to, in our pilot study in 2017, we're able to embed into WebMD and then to reach a more global population now. So one of our partners is Philips. We embedded in the Philips pregnancy app. So using the platforms that pregnant people already frequent and embedding whatever clinical trials, whatever studies that you're using or conducting into those where you already have a captive audience. The Penny Saver, the digital version of that, I think using those mobile applications is key.
And Venkat, given that you like I are pharmaceutical science professors, at one point I was in my life, but not interacting directly with patients per se, I'm curious, how do you get access to a community of patients that you can actually work with on your own research?
Well, even though I was trained as a basic scientist, my interest was to do clinical research and clinical work, and that's why I'm doing a lot of clinical research. So I do go and see the patient. In addition to the pregnancy work, I do organ transplant work and significantly involved in interacting with the patients since 1982-83 itself. And so in some ways, my path has been very different than a traditional basic science in pharmaceutical sciences. And so whenever there is a study in pregnant women, I go and see them. I talk to them because I want to know what makes them participate in research studies and what makes them not participate in some of the research studies. So I talk to them about their family. I talk to them about their babies and try to understand and learn from them. And so in that context, I do interact with them, and I really enjoy that interaction. And I think if we really want to make an impact, we need to interact with our patients as a human being, and we need to provide them the data that we collect. We are ready to publish things, but we are not as ready to share the data with our patients.
And I think that's a great point that you make. And I think we sometimes think, well, are they going to be able to ingest this information? But then, part of the challenge, and Lachey, I think you spoke to this in your own, in the Powermom platform, is meeting patients where they are and putting it into the language that patients can understand and comprehend. And I think that's an important aspect, especially to the point, Venkat, you made of how do we take these insights and actually turn them into actions that people can live and use and have impact on their lives. Wei, I'm curious, how does that translate into the registry and into the safety data? Does FDA look and see where are these signals coming from and try to look for trends that sort of point to places to maybe probe further, or is it really all just passive collection?
Are you asking me? Yes. So FDA does have different systems, passive surveillance, active surveillance, monitoring the safety signals from different sources. And then determination will be made, whether and how we follow up on those signals. So that's my short answer. Mm-hmm.
Great, thank you. Other questions in the room? We have one back here. Go ahead.
Sorry to get into the minutia, but I'm really interested in data standardization, and I'm wondering in each of your different platforms how you classify the maternal entity, the fetal entity, and then the exposed entity, whether it's the infant or the recipient of the breast milk, and how you suggest we as a community continue to term those entities systematically so that data capture can be shared between platforms.
So I'm not sure I totally understood the question, Ilona, but are you asking who we consider the participant for the breast milk biorepository, or?
Yeah, so for example, in our maternal studies, we link any safety data that we collect on the mom to the infant at the time of birth. And so we actually start a safety record for the infant at the inception of the study through our safety database. Okay. And I'm interested in whether or not you do a through line in these various different platforms and how you report that out. I was just talking about it, so for example, enlarged ventricles on an ultrasound, are you listing that under the mother, under the infant, and then when or the fetus, I should say, and when the infant is born, how does that reflect in sort of the capture of data systematically?
Yeah. So, the the enrolled participant is the mother, and then she's providing permission for the baby, once the baby is born, to be an individual that's followed as well. So an ultrasound finding we would report under the mother as a potential outcome for that pregnancy. But then when the baby's born, if the pregnancy ends in a birth that we can look at the baby or the products of conception, then whatever is captured to confirm that ultrasound finding is associated with the infant. So the infant would be the outcome. And same with breastfeeding studies. So, we're recruiting the participant as the mom or the breastfeeding individual who's providing us with the clinical data about her exposures and providing the milk sample. But the infant outcomes, we're capturing information from her about infant adverse events. We're capturing the neurodevelopmental information about that child and growth about the child. So the child is a participant as well with the mom's permission, if that makes sense.
That's also consistent with us. I will say birth outcomes, however, for example, small for gestational age, large for gestational age, we categorize that under the mom information. But if there's, for example, something abnormal that comes out of the hearing screen, then that's going to be categorized under the baby's information. But that's a great question.
And I think maybe, Alona, you're referring to one of the challenges you have if you're going back in EHR records is that, mom is dissociated from baby because of HIPAA requirements. The one place where we found that that's not the case is the UK because of everything sitting under NHS. And so actually the UK and CPRD, which was out of the UK regulatory agency, MHRA, I know that was a lot of acronyms, apologies, actually has that data linked. And so it's a really robust data set to actually explore that. But it would be better if we could do that here in the US, and it's a constraint. So it's nice these platforms are able to do that in a way that the normal medical establishment in EMR doesn't do. Yes.
Yes, I have a question regarding maybe ethics, responsibility, concerns, and dilemmas regarding, let's say, you have a patient or a person in your registry with a condition that needs specific procedures, diagnostic procedures, therapeutic procedures, and she has no access to that. How do you deal with that? How do you deal with that situation?
So a very good question. And since we enroll people, throughout the country who might live in rural areas or might not have access to services they could get at a large academic center, the study obviously is observational and not interventional, but we do serve as a resource for participants. So the person who's following that pregnancy, if they're looking for where can I go or who can I contact that might be able to, provide me access to some additional services that might be sought by that person, then we can help them try to do that in the locality where they live.
Yeah, that's consistent with ours as well. It's actually a question that came up within our IRB process as well. We are observational and interventional study, and we're able to capture this information, but we don't act on it. What we do within our platform, however, is have a host of different resources that the participants can access themselves. And the fact that we were able to show, for example, when they enter their blood pressure information, or we give them a blood pressure cuff, if they see, because right now we have guidelines, if they see that their recordings are outside of normal, what is currently normal or outside of their normal, they're able to then at least try to access a healthcare provider and say something's different. So it empowers them with their data, but we don't actually act on their information they provide for us.
If I may, a follow-up question regarding. Thank you for the answers. I'm thinking that maybe actually that data on lack of services or needed services is also important as the data that you have on conditions and diagnosis and outcomes.
Absolutely. I'm sure Tina does the same thing, but we also collect zip code data so we know kind of where the healthcare deserts are. And as we work to improve the services, which is why I think it takes a collaborative. And I think some of the one of the speakers said it earlier, we share our data, right? We can't make an impact if we just hold the data for ourselves. If we're going to have this large repository, we have to work with those who can impact the change directly, work with our OB colleagues, work with the legislators and different pharmaceutical companies to access data, identify data so we can make that difference. But I think it's collecting that data and how we share it is important as well.
Great. Well, we have basically run out of time, so I want to thank our panel for meeting with us. I think one of the things that we are really excited about in this area, and you heard from the panelists, is some creative ways to think about gathering information. That also is true in the biological samples. I'm sure that Venkat does what I used to do in the neonatal intensive care unit. If you realize that a lot of blood gets drawn from neonates and it gets thrown in the trash, with a good analyst, good capability to do drug analysis, you can actually turn it into value. So being creative is a real key component, and you'll see hopefully that these technologies continue to evolve and help us fill in the gaps. So thank you.